2.1 Amalaki, Philanthus emblica L family, Philanthacea. Amalaki sin. Emblica officinalis Gerten, a small or medium-sized tree, found both in natural state and mixed deciduous forests throughout the country ascending to 1,300 meters hills. Cultivated in home yards, gardens or grown as a roadside tree. Other names, Sanskrit, Amraphala, Amalaka, Datraphala. Assamese, Amlaku, Amlaki, Amlaku, Bengali. Amla, Daughtry, English. Emblic Myrabalan, Gujarati. Ambala, Amala, Hindi. Amla, Aimla, Kannada. Nalakai, Bella Nelli, Potted in Alake, Kashmiri. Amli, Ambali, Malayalam, Melika, Marathi. Anvala, Avlakathi, Oriya. Ainla, Anala. Part used. Dried pericarp, fresh fruit. Major chemical constituents: ascorbic acid, gallic acid, chebulinic acid, chebulagic acid, ellagic acid, 3-ethyl gallic acid, correlagin. Properties and therapeutic indications of dried pericarp. Ayurveda. Guna, ruxadry. Laghu light, rasa. Amla sour. Madhura sweet, tikta bitter. Virya, sita cold. Vipaka, madhura sweet. Karma, tridosajit pacifies 3 dosa, versi aphrodisiac. Therapeutic indications: Raktapitta bleeding disorders, Amlapitta hyperacidity. Important formulations: Thiovanaprasa AFI, Part 1, 311, Ravandasava AFI, Part 1, 1-4, Gasimularis AFI, Part 1, 118 Trifalacarna AFI, Part 1, 715, Trifalati Tela AFI, Part 1, 821, Doctralaha AFL, Part 1, 21-2, Pharmacological study. Alzheimer's disease. Administration of Amalaki Rasayana and Ayurvedic formulation showed improvement in learning, memory and neurometabolism in APPPS1 mouse model of Alzheimer's disease AD. Amalaki Rasayana increased the 1,3-C labeling of amino acids, suggesting a significant enhancement in glutamatergic and GABAergic metabolic activity in ABPPPS1 mice, similar to that observed with donepezil, a standard FDA drug for treatment of AD. These data suggest that AR has potential to improve memory and cognitive function in AD. Clinical study. Administration of Amalaki Rasayana for 45 days maintained, enhanced the DNA strand break repair, DSBR, in aged individuals without any side effects. The individuals with different body mass index showed differential DNA strand break repair capacity. References: 1. Quality Standards of Indian Medicinal Plants, Volume 8, New Delhi. Indian Council of Medical Research, Ministry of Health and Family Welfare. Government of India, 2010, pp 160-171. 2. The Ayurvedic Pharmacopoeia of India, Part 1, Volume 1, New Delhi. Department of Indian System of Medicine and Homeopathy. Ministry of Health and Family Welfare. Government of India, 1986, pp 5-6. 3. The Ayurvedic Formulary of India, Part 1, Second Revised English Edition. Department of Indian System of Medicine and Homeopathy, Ministry of Health and Family Welfare. Government of India, 2003, 4. Tuwarivi, Sabake, Viraya P. Jose J. La Kosha SC, Patel Ab. Amalaki Rasayana improved memory and neuronal metabolic activity in ABPPPS1 mouse model of Alzheimer's disease. Journal of Biosciences 2017, 40-3, 363-371. 2.2 Apergita, Clitoria ternidia L. Family, Leguminosae, Apergita clitoria ternidia L. A perennial twining climber commonly found all over the tropical parts of country, being cultivated, and also found wild, growing over hedges and thickets. Other names: Sanskrit, Urakarni, Asphoda, Vicacrunta. Bengali, Apergita. English, winged leaf clitoria. Gujarati, Garni. Hindi, Apergita. Koil, Kannada, Girakarnik. Malayalam, Sankpushpam. Marathi, Gokarni. Oriya, Apergita. Telugu, Dintana, Sankapushpam. Part used. Leaf, root. Major chemical constituents: apergidin. Properties and therapeutic indications of leaf. Ayurveda. Guna, Laghu light, rasa. Tikta bitter, Katu pungent. Kasaya astringent. Virya, sita cold. Vipaka, Katu pungent. Karma, Pittahara pacifies pitta. Kanthia wholesome to throat. Visahara antidote. Therapeutic indications: Custa diseases of skin, Nutridosa urinary disorders, Vrana wound, Visa poison. Important formulations, Bhattaraktantaka AFI, Part 1, 2044. Pharmacological studies. Nootropic activity. In diabetes-induced cognitive decline rat model, a phenolic extract of C. Ternidia leaves CT at 200 and 400 mg per kilogram significantly increased spatial working memory, spatial reference memory and spatial working reference in retention trials on Y maze, Mars water maze and radial arm maze respectively. Further CT showed decrease in the acetylcholinesterase activity, lipid peroxide, no and increase in SOD, CAT and GSH compared to diabetic control group. Aqueous root extract of Clitoria ternidia, 100 mg per kilogram BW, when administered in young adult Wistar rats for 30 days, showed increase in passive avoidance learning and memory, dendritic intersections, branching points and dendritic processes arising from the soma of amygdaloid neurons. Clinical study, antioxidant activity, in a randomized, crossover trial. Clitoria ternidia flower extract CT evaluated for its effect on postprandial plasma glycemia, response and antioxidant status in healthy men. Treatment with the beverage prepared with 1 gram of CT in 400 milliliters water increased plasma antioxidant capacity without any hypoglycemia in the fasting state. The drug also found to improve postprandial glucose, insulin and antioxidant status when consumed with sucrose. References. 1. Quality Standards of Indian Medicinal Plants, Volume 4, New Delhi. Indian Council of Medical Research, Ministry of Health and Family Welfare, Government of India, 2006, pp. 84-291. 2. The Ayurvedic Pharmacopoeia of India, Part 1 Volume 4. New Delhi, Department of AYUSH, Ministry of Health and Family Welfare, Government of India, 2004, pp. 8-9. 3. The Ayurvedic Formulary of India, Part 1 Second Revised English Edition, Department of Indian System of Medicine and Homeopathy, Ministry of Health and Family Welfare, Government of India, 2003, 4. Talpate Ka, Bosalue, Zambari Mr. Somani RS, Neuroprotective and Nootropic Activity of Clitoria ternidia Lin, Fabaceae leaves on, Diabetes-Induced Cognitive Decline in Experimental Animals, Journal of Pharmacy and Bioallied Sciences, 2014, 6-1, 48-55.